I'm live streaming from some tank water, from the fish tank, and we have found something interesting in our tank water at school, so I'm gonna share with you. I'm looking through a stereo microscope. Ciao Big, che faccio? Studio microbiologia o guardo la live sui batteri? Well, why not both? Entrambi direi. 9 persone. Allora, qua sì, non è proprio microbiologia, però sono animali piccoli, quindi di fatto è microbiologia. Mi sono entusiasmato perché ho trovato delle idre. Let's see if I can show you some hydras. It's on a different level. We're gonna get down here. This is one of our small hydras. Is it on focus? Okay. Can you see it? Sì, oggi live in inglese. Because I want to meet the international public. La musica si sente? Can you hear the music? Is it too loud? Is it too low? Ah, un fastidioso rumore grigio. Così? Magari è questa canzone, eh? Proviamo a cambiare. Così si sente qualcosa? Io sento una canzone col pianoforte. Così? Ancora fruscia. E così? Così si sente ancora il fruscio? Perché magari il fruscio dipende dalle mie cuffie. Prova, prova. Can you hear me? Let me know if you can hear me and if you hear a music or some noise. Ok, so you still have noises, c'è ancora fruscio sotto? Fatemi sapere se migliora o se continua a fare rumore. Ok. Allora, per il fruscio non so cosa fare, perché per ora ho solo questo microfono qua. Vediamo un po'. Questo non va bene. Ok. E prova a ridurre il gain. Così? Meglio? C'è ancora fruscio? Grazie, Ciao Big. Grazie anche Mr. Gimsar. La vedete la mia idra? Idra funesta. My small hydra. I'm trying not to move the microscope and the table, but it's not easy because everything is on a very trembling table. Everything is trembling and moving. And since the magnification is quite intense, okay, you should have the music like this. Can you hear both me and the music? Let me know. Music is quite low, huh? It's just like an ambience music. Okay. Oh, here we have another hydra. Let me move it. Move it. Oh, here it is. This one is bigger. Look, isn't it cute? Molto carino. Oh, it's difficult to move things under the microscope. Certo. Questo è uno. This is one. And who's there? 2 new followers. Awesome. Thanks. I'm trying to focus on my second hydra. And here she is. This one is very long. Do you know hydras are fascinating animals? You know why? Because they have like an infinite ability to regenerate. So if you cut an hydra in 2, in a few days you get 2 hydras. Come mai sono così particolari? Particolari perché appunto sono capaci di rigenerarsi all'infinito. Sono non solo eucarioti, sono proprio animali. Sono animali pluricellulari. Il campione arriva da un acquario, acqua di acquario dello Zoo Megastore, che ci hanno dato. Poi lunedì forse ci fanno avere degli altri campioni. Vediamo se riesco a mettere a fuoco. Più di così no. Possiamo andare un po' più giù. Sono praticamente dei parenti delle meduse. Hydras are related to jellyfishes. And I'm citing from Wikipedia. It's a genus of small freshwater organism of the phylum Cnidaria and the class Hydrozoa. And in the phylum Cnidaria you also find jellyfishes. So you have the jellyfishes, le meduse, which are in the same phylum, and in fact their behavior is very similar. They have little stings, the nematocysts in their tentacles that can be used to catch prey by stinging it very very fast with poison, and then when they got the prey, they uses their tentacle to convey it to their mouth, which is the only opening they have to the surroundings, and they can eat it entirely in its entirety, and they will then release some enzymes to digest the prey. Super cool. They're so tiny. In this case, you see a magnification of about 2 by 20, 30 times, plus the magnification of my cell phone and your screen magnification. What do they eat? Cosa predano? Ho visto un video l'altro giorno su YouTube dove mangiava una larva di zanzara intera. Se l'è messa proprio dentro completamente. I've seen a video about them eating a mosquito larvae, which is quite big for them. And I think they can also eat smaller things, but it's the first time I see them in real life, so I'm trying to understand how they work. It's completely new to me. Let's see if there's something in Wikipedia on it. What do they eat? Let's have a look on Google. Hydra. What do they eat? Crustaceans. Hydra are small carnivorous and feed mainly on small crustaceans like water fleas, the Daphnia, and small worms. Although Hydra are fairly simple animals, the stinging cells which they use to catch their prey are quite complex structures. Ok, quindi piccoli crostacei come le Daphnie, le pulci d'acqua e piccoli vermi. E io cosa gli do da mangiare? Ho dei nematodi che posso dargli da mangiare. In un altro campione, che adesso magari vi faccio vedere, ho i nematodi. I have nematodas in another sample, so maybe I can get some of the nematodes and give it to the Hydra. Ciao Archeo friendly. We can use them. Yeah. La particolarità delle idre, come dicevo prima, è che non invecchiano mai, hanno questa capacità di rigenerarsi all'infinito e hanno la non senescenza, quindi non invecchiano, le loro cellule sono sempre giovani e fresche. They don't get old. In the same water sample, I also find some planarias, which are flatworms, and they are super cool too. They are like a worm, but they are flat and very small, like less than 1 millimeter. And their peculiarity is that if you cut them into pieces, they can regenerate the other piece. So if you have a planaria with the head and the tail, you can cut them in two, and you have two viable parts of the planaria, and the head part will regenerate the tail part, and vice versa. So if you have the tail, it is still living, and in a few days, like a week or so, it will regenerate the other part, the missing part, with the head and the eyes and everything. And they're super cool too. I'm not sure if I have some planarias in this sample, because it comes from my school where I teach. So I got some of the water to take it home and analyze during the weekend, but I don't know if I have taken some planarias too. But for sure we have hydras. I had like 3-4 hydras, and in this sample you have at least 2. Maybe we're gonna have some look around also with the other microscope, the optical microscope, because with the optical microscope everything is cooler because you see with a greater magnification. And well, maybe let's move to the optical microscope in a few minutes. So this is just how the hydras are in like 30 time magnifications. This is one, and there was another one which I can't find now. Where did he go? Oh yeah, this is it. This is the second one, the smallest one. As you can see, it is attached to the bottom with its tail. Let's call it tail. Here it is. It's attached to the bottom. It's like sticky. Here you can see it better. Okay, it's moving because it's alive. It's so cool. It's really the first time I encountered this kind of animals. Wow, so cool. I studied hydras when I was when I was 16, 17, when I was in high school. But I never expected to find them so easily in water and tank water. It comes from the fish tank in a zoo store where they sell animals, fishes, and also water for the fish tank. And they gave us some water samples to take it to school and analyze it. And I brought some home for the weekend in order to get further analysis, and it's so cool. Hydras. Hello guys, hi Chiara. Welcome to my stream. I'm trying to stream in English, as you have understood, because I'd like to know if someone is looking us from the US, maybe from other countries where they speak English, and maybe they're interested in science too. So since we're streaming in Italian all week long, I decided to start with some streaming in English, with the help of my microscopes and with the help of these tiny little bugs you can see here, like my hydra, the first time I see an hydra in my life. I was super excited yesterday when we discovered hydras. The lab technician called me, Professor, come here, have a look, and I was looking at another sample, and then I saw something moving. It was like a worm, and I was, who are you? And I followed the worm, and then I found it was the tentacle of an hydra, and I was like, what? This is an hydra! First time I see it. And I was super, super excited. So I wanted to share with you because it's so cool. What do you think? Do you like hydras? It's like a jellyfish, but it lives in fresh water. And it's smaller, way, way smaller. It's really useful also for Italian followers. I hope so. Do you enjoy English streaming? It's like studying English. Sorry for moving the microscope, but each time I touch it, everything moves, because it's like, in water, so everything moves, and it is magnificated many times, so every little movement I do is made bigger, made movement bigger again. So pretty, yeah, they are. As I told before, I also had some planarias in this sample of tank water, but I'm not sure that I brought home some planarias because they're so tiny, so small. In this case, because there are also other versions of planaria which are bigger, and this one is so tiny that I barely saw it with my naked eye. So I just went with the pipette and I took some water samples and I brought it home. And maybe let's have a look with the other microscope. Let me set the microscope so that you can follow me in setting the microscope. I brought some here I am with my cell phone attached to the stereo microscope. Now I remove it and I attach it to the other microscope, the optical microscope. And in a few seconds you should be able to see things. Now let me just do like that. Meanwhile, I show you how I prepare for the microscopy. Here I am. So let's turn on the microscope. Is it plugged in? No, it's not. Gotta plug in. Be right back. Here we are with the other microscope. Let's move it. No, I'm not gonna move the hydras because I have to take it from the sample and put it on the slide. So I need. Where's my slide? They're so transparent I can barely see them. Okay, this one's cool. A little bit. Here I am. Okay, so microscope slide. I'm gonna use my stereo microscope to catch the hydra and to put it on the slide. I hope not to damage it, although they have a super regenerative power, so they're really able to regenerate themselves after they have some damage or after they get destroyed in multiple pieces. Like for instance, I read that the other day that some scientists cut hydras in multiple pieces. They broke them completely into cells, they centrifugated them, they spun them, and after that they regenerate 2 hydras, not 1. So it's super regenerative powers. Okay, let me catch these hydra. Not easy to work under the microscope, but I'm trying to do that. Maybe some more light, like this. Perfect. 1, 2. Here you are. I'm gonna free them. I don't wanna kill them, so I took the 2 hydras in a very, very small water droplet. And let me see if there is something. Did I take it? Or did I not? Oh, maybe I didn't. I can't see it. Nope. Where is it? Is it in the pipette again? Let's do it again. I thought I took them, but I didn't actually. Let's try again. Now I have to find it. Maybe you can see while I take the hydras. I reduce myself again. I put me here, and let's detach it, and I'll touch it again. Okay, so we can see more or less the same thing. yes, now you see the same thing. Also because now I'm watching through the telephone. So okay, let's find the Hydra. I hope not losing. Did I lost it? Okay, here it is. Here you are, darling. Come with me. Do you see? Okay, here it is. It's on the bottom. Tightly attached to the bottom. Here's my pipette. Oh gosh, but this pipette is too big. Maybe this time it worked. I got an excess of water that I put back to the microscope, and now let's check if it is in the droplet I took. Oh, here it is. And let's see. And I don't see it. Really? No, there's nothing. Where did it go? They keep running away from me. Absolutely no hydras whatsoever. Remove some water. Okay. Are you a hydra? No, you are. Oh, here it is. No, is it? No, it's not. Or is it? Let me zoom in. But it doesn't look like an hydra. Okay, so where did it go? It's impossible to catch it. Sorry for the light. It's not easy to move and set the light properly and to look for the hydras as well. Nope. There's nothing here. Did it stay inside the pipette? Or did it stay inside the water sample? Just to be sure, I washed it. Let's check if there's nothing here. Okay. Okay. Nothing in here. So I can clean it again. Let me see if I can find something. So difficult to do that. I thought it was easier to take it with the pipette, but actually it's not. Here it is. I can see it with my naked eye. Did I take it? Okay, now I have it. I'm sure that I have it. I can see it with my naked eye. I'm gonna show it in under the microscope. Can you see it? Oh, okay, now you see it. Got it. Kid for such a nice music. Thanks. Hey Wally, where is Hydra? Yeah, I'm Wally today, as you can see. She's quite suited for what I'm doing. So looking for hydras. Okay, I hope she's still alive, but they're like immortal, so she's probably alive. Okay, let's move to the other microscope. Let me check the chat if you wrote something. Okay, no, I'm up to date. So let's put another little bit droplet of water. Here we are, because I don't want it to be smashed by the weight of the cover glass, which is quite thin, but you know, it's still something gigantic I'm putting on it. Take a clean one. Oh, 12 cover glasses. Okay, I'm gonna put the cover glass on it, and let's have a look at it with the other microscope. I'm gonna switch off the light here. Let's move to the other microscope. Come with me. Here to there. I have to remove the other samples because now I have some blood samples on it. And now I'm gonna perfect the image. Okay, don't need this anymore, don't need this anymore, and this too. There's a sample here. Okay, the blood samples. And here's our hydra on a glass. Okay, so I can remove this microscope. And here we are. Okay, let's have a look at these monsters living in pond water. Okay, so let's start with four. Here we are. Are you ready? Webcam. Just do and here I am. Maybe I'll also cut the webcam like this. Since I'm not that interested. And maybe too much. Let's cut this. Expand it. Okay, awesome. Good. And now let's try to focus it. Here it is. Hi. Hi, little hydra. Okay, let me just set the light because I have to move my diaphragm and to open it a little bit. Okay, cool. Now we should see it quite well. And here we are with the hydras. This is 40 times magnification plus the magnification of my cell phone and your screen. And here, see my tiny monster. Look how cute it is. Hi Hydra, how'd you do? So cool. It is so cool. Look how it's expanding. Now it's unwinding because she was stressed by the movements from a container to the slide, and now it's relaxing, it's unwinding, and it opens up. And maybe this music is suited for this moment. It's the cousin of jellyfishes, same phylus. Isn't it nice? I'm gonna leave it for a few minutes in order to explore the new world where I put it. So I mean the glass, the microscope glass where I put it. I'm gonna get the book that I used when I was in high school, in which we can see some more information about hydras. I'll be back in 30 seconds. 29, 28. Here I am. Spectacular, and this music is perfect for the video. I agree with you. Okay, here I am with my 2 books of biology that I used when I was in high school. Pervs, Orions and Heller biology course. Ooh, where is it running? Don't run away, come here. Ciao. So cute. Okay, let's find the hydras, because I remember studying it when I was in high school, and it should be in the 2nd book, but I'm not sure. Have a look at the index. Hydra. It's hydrozoiset. Yeah, it's on the 2nd book. The book with all the worms. Class of Hydrozoan, the only group in the phylum that includes also freshwater species. They have the form of a jellyfish. Vad är 24? Flatworms Okay, I also have flatworms, but in the other sample. That side. Area. Hydras up. Mm-hmm. Cnidaria. Phylum. Cnidaria. Tentacles. With epithelial cells with muscle fibers that allow this animal to move, and some neuronal networks. So they also have a nervous system that coordinates the various life activities. They have only one mouth opening. They don't have an anus, so a digestive tract. They only have a mouth, and everything happens in it. So when they're done with their food, they just have to eject it from the same spot where they got it. The anus and the mouth coincide in this kind of phylum. I hoped there was more than this, but there's nothing more. No, there's nothing more. I remember studying something more about hydras, but I didn't. Okay, so tell me something about you. 10 people watching. Awesome. Do you want to write something to tell me something about you? Did you have any experience with microscopy or exploring the microworld somehow? Maybe in school, maybe at home, maybe just with a magnifying lens. Let me know who you are and where are you watching from. While we watch this hydra. I'm really curious about cutting it into pieces in order to get more hydras to make it regenerate. And also to see it regenerating, so maybe like cutting it in two and getting two hydras. That would be cool. I was talking about hydras today with my mom while we were eating pizza, and I told her, what if the pizza also auto-regenerates? Maybe you can eat like 90% of a pizza and then keep the last slice and put it in the fridge, and then it regenerates again, and in a few days you get another full pizza. Isn't it like wonderful if it could happen? I'm a pizzaholic. For those who knows me, I'm definitely a pizzaholic. Gothic me is our last follower. Don't be shy, write something in the chat. Let me know that you're there, not just watching but also listening to me. Let me explore if we have something else in the same sample. Okay, we have some weed here. is. Let me see what's here. Also, it's just grass. But now we're gonna get some closer, not just to the hydra. I mean closer, especially to the. Oh, so cool. Especially closer to the grass because maybe we have some luck and we can find something in it. Okay. Let's have a look. Here we are. Now 100 times magnification. And let's see what's in here. Light and more light. Is there something cool? It's just algae. Is it everything? Maybe 400 magnifications. Okay. We have green algae, brown algae, I'm entirely another word that just graduated civil engineer in search of first work. But I really like new things and listen to the vulgars. Thanks, Mr. Kimsala. Grazie. Ingegnere civile. What kind of work are you looking for? Oh, these are some green algae. Can you see it? The brown algae, different color. Try not to move it a lot, but it's difficult. Tiny animals feeding, not animals. Single-celled organism. Here we have algae. I wanted to show you some vorticellas, because we found some vorticellas and they were amazing. Let me see. Wow, you can really see everything inside this green algae. You can see the inner organelles in the cell. It's incredible. I really love microscopy. Having the possibility to explore other worlds that are under our eyes, but we don't see them because they're too small. Awesome. It's awesome. Okay, let's go to the other side. Let's go to the other side, where we have other grass. And here it is. Maybe there's something moving. And nope, there's nothing here. Not even a vorticella. But we have some single-celled organism here. Very shy, moving. Can you see it? It's very faint, but yeah, maybe you can see it. Let's have a look. Oh, something moving over there. Who are you? Come here, show yourself. Are you a vorticella? Let's wait for a few seconds. They're very shy. They tend to move when they feel something moving, but it's not the gel. Okay, let's change the magnification to 40. Who are you? Let's have a look closer. And here we are with a closer look. Who are you? And what do you do in your life? Oh no, it's not a vorticella. Is it an euglena? Oh. Could it be an euglena? That would be awesome. Oh yeah, I bet you are. I can see the flagella. The chloroplasts in it. Ooh, so tiny, so cute. I try to follow it without losing it. It's not easy because it moves on different planes, different layers, and I have to change focus constantly. Yeah, but it is. But you're so cool. Come here. Don't be shy. So cool. Something that will unite my passion, programming, public transport management, is interesting at the moment. Cool. Where did you study? Which university did you graduate from? So many things that move. I bet they are bacteria. Let's have a look at our hydra to see it if everything is good. Where did it go? Where did it go? What's that? Is it the hydra? Oh gosh, it's so big! 400 magnification. 400 times magnification. Look how big it is. It's like the kraken. You can see the segments in its tentacles. And inside these tiny tentacles you have the toxins. The cnidocytes that contains like an harpoon. Their weapon. It's like a needle they used to inject their poison to paralyze the animals, their prey, and then they use their muscles and nervous system to attract it to their mouth. You're having a close look at the hydra, and this is the head, the mouth on the head, if you can call it a head. Wow, I'm amazed. Really like it. It's gigantic. I didn't expect it to be so big. Let's have a look from the distance. Wow. And it keeps moving. I'd like to know what to feed them. Look how it drinks. completely shrunk. It's so funny like this. It's super cute. Can you imagine painting 2 eyes and make it like the funniest octopus? Or in this case, 6-topus, because it just has 6 tentacles. From Padova University, Padua. Great. The poison has some time of effect on humans. I'm not sure, but they're very, very tiny, so probably they're too small to have any effects on us. But since they're related to jellyfishes, if jellyfishes can do that, and they have the same kind of cells, the same kind of poison, let's say, probably it's just the number of tentacles and the number of cells that touch you and that sting you. In this case, maybe they're too small to have an effect on humans, because maybe they just can sting you with a dozen of these cells, and maybe it's too little to provoke any response. But I don't know, I'm not sure, I don't want to try. I've never been bitten by a jellyfish. Did you? Did you ever experience that? Being bitten by a jellyfish. Being stung more than bitten. Let's try it. Let's just enjoy some music with those images. Let's make it an ASMR live. Also, Andrea, Kid for Sacha, asks, hey guys, where are you from? For those who doesn't know me, I'm Roberto from Italy, and with a group of friends we are streaming on this channel, mainly in Italian, and we try to disseminate some science. We talk generally about biology, neurology, and diseases. Like yesterday night we have a live on sexually transmitted diseases, and what else? Archaeology and iconology, so the study of ancient images in manuscripts or in statues, in order to understand the context, understand the history directly from the source, from the image. Luckily, never been bitten by a jellyfish. Lucky you. I heard it's quite painful. I never tried it, luckily. Who knows about this hydra? Maybe it's feeling comfortable, maybe likes the music. It has unwound. You see how long it is? It's very long indeed. And I think the other one was bigger. And if you also expand the tentacles, it's like super long. Okay, time for the hydra to go back to the pond. I mean, I'd like to show you some other samples, other living organisms from this sample. Maybe I'll just try to put the hydra back. Oh, she's so tiny. Ciao, hydra. Bye bye. I'm gonna put you back. Okay, here you are. I'm trying to put the hydra back without damaging it, but also you know that if I damage it, it's not going to be a problem because they regenerate. But I try not to damage it. It's better. Where is it? Okay, it's stuck to the glasses, so it's not here. Let me be sure it's not here. It's not. Okay. And I try to wash it, or maybe I have made a very tiny pipette. I can show you the dimensions of this pipette. Look at how small it is. I try to put it under the microscope. Okay, here's the hydra. I don't want it to get dehydrated. I'm trying to touch it with the tip of my pipette. Oh, it's not feeling good. Look at the tip of the pipette. Look how small it is. Very, very small. Okay, so now I try to use it to bring some water back to the hydra, to rehydrate it, and to detach it from the glass and put it back. Are you back? Seems like it's back in the water. Yeah, let me check if it is not here. Okay. No, it's not. Oh, let's see, it's still here. Okay, so I got to convince her to come with me to the water again. Come with me. Come with me. Let's go back to the water. Maybe if I use some water to wash it, like this. Okay, let me see. There should be anything. Just water droplets, okay, that's good. But no hydras at all. Here. No hydras here. Okay, so perfect. I managed to put it back inside its glass. Good. Good, awesome. If dehydrated, can it survive? I don't know. I know it's an animal that lives in the water, so probably it needs water to stay alive. And also its prey are in water. But I mean, if it's dehydrated for a very short period, it's true that they are tough animals. They're very resistant, but without water, maybe it just collapses or the cells die. I'm not sure. It can die, but its peculiarity is the regeneration properties. It's not immortality, so it can die. But if you cut a piece, it can regenerate. And from the piece you cut, it will regenerate another animal. So let's take another sample from this pond water. And I try to show you some vorticellas. They're very cool. Also with the optical microscope. Let me see. I just try to fish one very small plant of water lentil. And here we are. And Let's hope there's something attached to them. A few drops of water, and here it is. Cover glass. Let's have a look. Come with me to the microworld. And here we are. Oh, we're lucky. We have something interesting immediately visible in our specimen, which are the vorticellas I was talking about. Now we get a closer look. Let's just explore a little bit. You see the stems of these tiny plants. They're water plants, not bigger than 2 millimeters. We have a crescent moon shaped green algae here. And here's the plant I was referring to. You can see it's very thin, so you can see the individual cells. Okay, let's have a look at the individual cells before moving forward. And here we are. 400 magnification. You can see the cells. These are plant cells with the chloroplasts in it. Isn't it cool? Maybe is it too much light? I can reduce it a little bit. Or I can try to move the light from here. Okay. So you can see the plant cells rounded with the green chloroplasts in it that makes photosynthesis. And the unicellular organism moving around. Okay, but let's go to the edge of these plant cells, because most interesting things are found at the edges. Like this one, it's probably, it looks like a blue algae, but it can also be a cyanobacteria. They make very long threads, like this one. Well, here we have bacteria moving. Can you see their movement? They're so tiny. 400 times magnification in here. Very, very small. But we have them. Let's have a look over here. This is still the plant cell, okay? Let's go to the other side. Do we have here? Bacteria again. Bacteria are everywhere. They colonize like every ambient, every part of the world. Here we have some green algae. You can see the cells are divided by a cell wall. So every individual cell is like a tiny box, but they make chains, very, very long chains. And you also have chains of bacteria, but they're the tiniest one, the smallest one. Okay, let's go find some vorticellas. Oh, here we are. Here is a vorticella. I was talking about you. Can you see how fast it moves? And now we can see or all the cilia, it's like eyelashes they have on their cone, on their bell, and they used to this kind of eyelashes to move the water around them. You can see how the microorganisms are moving, are flushing around them because they move the water and they create a vortex. This is why they're called vorticellas. And they feed with this microorganism. You can also see a gigantic vacuole filled with water. It's like here where I am. I'm pointing not that way. This is the gigantic vacuole. Oh, it's difficult to point it. It's transparent, it's translucent. It's filled with water, and it's contractile vacuole. It fills with water by osmosis, and then it expels the water outside. Yeah, it's cool, isn't it? Vorticellas. And when they feel menaced or threatened, they immediately shrink their stem, with which they attach to the bottom of the container or to the plants, like in this case, and they hide themselves. So it's like a spring, and they can shrink it or expand it. I've seen something moving here. What's that? Another vorticella, probably, on a different play. Yeah, it's another one. Hi, 2 vorticellas in one image. This is 400 times magnification. You can see they're filled with bubbles. All these bubbles are food vacuoles. So they ate microorganisms, and now the microorganisms are trapped inside these tiny spheres, the food vacuoles inside the cell, and they will be digested by the enzymes produced by the vorticella itself. So they produce vesicles, bubbles, let's say, with enzymes. They engulf the microorganisms coming from outside, and the two vesicles merge in one big vesicle, which is the food vacuole, and they are digested. Look how they move. Okay, something is moving inside it. It's the new food vacuoles. It's producing continuously. So the tiny bacteria that are moving around the mouth, let's say, of the vorticella are getting inside, that they are trapped inside the membrane. It's a process called endocytosis. I could stay here hours watching vorticella. Look how everything moves. The new food vacuoles are taken from the surface. They are detached from the membrane and they are taken under. And look how it flashes. You can see the vortexes that it's generating. It's like making water circle around it. Let's try to change the lighting in order to see if you can see any of the cilia they're moving. It's difficult to see. Oh, sorry. I touched it. I touched the microscope and it it retracted immediately. As you can see, I'm changing the light because I'm taking the what's it called, the diaphragm and the condensator closer to the sample, and I'm trying to close the diaphragm without moving everything. And here we are. Diaphragm closing. Okay, good. It's not centered. The problem with my microscope is that it doesn't have a real diaphragm. I have another diaphragm I got from another laboratory and I try to attach them. It's not always working. Super cool. Okay, let's go back to 100 times magnification and let's see how the Vorticella look like. We have the two Vorticellas we were looking at, and let's find others. There were some here. Here we have some. One Vorticella here. A Vorticella here. Do you like them? I like Vorticellas a lot. They're very, very cool. For people watching, thanks for watching. If you want to tell me something about you, I'm curious. Where are you watching from? Or just tell me something curious about you. Your experiences maybe with the microworld if you had some. Look, we have two Vorticellas here. Oh, they're the same as yeah, they're the previous one. Let's go here. There was plenty of Vorticellas here. Here we have one. Here we have, how many? One? At least one, maybe two? Yeah, two Vorticella. This is the end of the slide, the microscope slide. Oh, do you remember there was a moon-shaped green algae? Let's find it. Where was it? I've seen it before. Let's go back and find it. How many Vorticellas? There are plenty of them. Plenty. Amazing. Look how many here. We have like 3 different Vorticellas in this image. Another one. Where is the moon-shaped algae? Here it is. Okay, I wanted to show you this in 400 magnification because it's cool, isn't it? It's like a green banana with tiny green dots in it. Unfortunately, it's covered by other stuff, but can you see it? Is it cool? The green stuff is chlorophyll. When I was younger, I remember, ciao Gabri. When I was younger, I remember seeing this kind of green algae before, and I also knew the name, which I don't remember now, but maybe I can Google it. Like, it's like a Selene something, moon-shaped green algae. It's called it is. Freshwater life. Here, this one. It's a desmid. Desmid. Oh, okay. Let me see. Freshwater life. Closterium. Closterium is the name of the crescent moon. It says it moves slowly by raising the back and then the tips. Oh really? Didn't know it moved. Closterium. I also had some diatoms because I remember seeing the diatoms. And now my biggest dream is to find a stentor, which is halfway between a paramecium and a vorticella, but they're bigger and they're evil. They're sting other microorganisms in order to feed on them. Who's there? Someone moving. Who are you? Hi. Why are you hiding? They look like paramecia. But they're hiding. Can you see something is moving? Green algae, brown algae, bacteria, and probably we also have some cyanobacteria. Okay, so I think that for this life it could be enough because I'm gonna explore more from this microworld in the next days. And okay, so thanks for coming. I'm glad you've been with me, and see you in the next streaming. Subscribe to our channel and follow us in order to know the next lives, in order to get some notifications when we are live. You can just push on the bell button. Okay, so thank you again for coming with me to the microworld, and thank you for watching. See you next time.